Malaria is a disease caused by plasmodium species spread to human beings through an infected Anopheles mosquito. Africa is a developing continent in which 90% of more than 400,000 deaths that happen in the world, as reported by the World Health Organization, happen here, and 61% of that number are children under the ages of 5. There are drugs currently being given to people who suffer from malaria. However, the plasmodium species has become resistant to such drugs, resulting into the need to develop new and effective drugs that can be used to eradicate this disease. In our study, we focused on a protein called type 2 NADH quinine oxidoreductase protein, which functions in the electron transfer from NADH to the quinine through the FAD cofactor. We used the South African Natural Compound Database, in which it has got 623 compounds, and we docked them to our protein. We then further selected the hit compounds or the hit ligands that interacted well with our protein, and went further to perform molecular dynamics to see how the ligands were interacting with our protein. We wanted to calculate the fluctuations of the ligand and to see whether the ligand was indeed in any way affecting the functioning of the protein. So in molecular dynamics, we had to imitate the exact environment of the protein as we are using bioinformatics techniques. We created a membrane using CHARMM GUI, and we embedded the protein, the NADH2 protein, into the membrane. And we also included our cofactors, the 2 FAD cofactors, and the 4 magnesium ions in each dimer, as our protein is a homodimer. We seek to see if our ligand will indeed affect the way our protein functions. We seek to hinder also the NADH from binding to the protein, so that when NADH fails to bind on the protein, meaning that no electrons will be able to be transferred to quinine to complete the cycle of protein pumping, and therefore probably result into the death of the plasmodium. Thank you.